honor to talk with you today. Thank you to the grad students for the invitation. And thanks for the Corteva people for providing support for this event. I really love the fact that grad students are responsible for everything involved in putting it on. So I have added Scott Healy as a co-author here. Scott is our applied wheat breeder, known to many of you. And I got some examples from him, so I thought he would be a worthy one to include. So what I intend to do is to provide some general concepts and perspectives on breeding for resistance to pests, and then give some specific examples of Colorado breeding programs, one for a disease problem, one for an insect. And I was very much guided by a couple of recent review papers, one on navigating complexity to breed disease-resistant crops by Rebecca Nelson and colleagues at Cornell, and the other one on resistance to insect pests by Angela Douglas, who's also at Cornell. So these were very useful for me in bringing me up to speed on what some of the literature is suggesting. So I'm not sure if this plant disease triangle was presented yet or not, but it's one of the basic concepts in every plant pathology course, indicating that there are three important points to produce disease, or disease resistance for that matter. So there's the host, the plant, a pathogen, and then the conducive environment. Now, those of us in the plant breeding realm focus naturally on the plant host, and maybe too much on the plant host without thinking about the other aspects. So there probably isn't too much we can do to influence the pathogen, but I think certainly we ought to keep in mind more the environment. The irrigation environment, for example, the fertility level, the crop rotation pattern, the pattern of diversity of crops. So let's see. So anyway, just want to make that comment just to provide some background that I think this whole field of breeding for resistance would benefit from looking more broadly at the agroecosystem and not just the plant part of it. So resistance to pests, I'm going to talk initially thinking pathogens primarily. And there are two types of resistance that are recognized, qualitative and quantitative. So the diagrams of the leaf indicating the qualitative, there are some completely resistant and others that are very susceptible. Whereas the quantitative is more of a gradation of phenotypes. On a population level, you can see 3-to-1 segregation indicating a major gene segregation as opposed to the normal distribution with quantitative traits. So at the genetic level, the qualitative resistance is typically, but not always, dominant. So we would have both the dominant homozygous and heterozygous condition being resistant, the recessive being susceptible. Whereas with the quantitative, we can see a range of genotypes for each of the genotypes. So just to review some of the other features of these different types of resistance, qualitative is considered vertical resistance, sometimes that's the term that's used. Controlled by major resistance are R genes, often involved in pathogen recognition. Distinct reaction types, pathogen race specific. And this is a really important point regarding durability, that the major genes are resistant against generally a particular race of the pathogen. So if there's a race change, that resistance may no longer be effective. Usually a high level of resistance, but less durable. So one consequence of that is that it's more suitable for annual crops that can be replaced more frequently, something like corn or wheat or soybeans, as opposed to grapes or apples, which are perennials. So the quantitative, sometimes termed horizontal resistance, controlled by many genes of small effect, and various mechanisms. There's the continuous range of reaction, not race-specific, so generally affecting basal defense mechanisms effective across various races. Often the resistance is partial. It's not a strong, high level of resistance. And it's considered more durable. But because of the number of genes involved, it's a more challenging breeding. The breeding challenge is a higher level. So with our gene, the classic situation is boom and bust. So this is showing the deployment of our genes over time. Initially, you might have a new cultivar that has a very effective R gene. And it might work for a period of years or even decades until the pathogen population overcomes that resistance, and then you have the bust. A new R gene might be deployed, and again, you have the same situation where you have a high level of resistance, which is then overcome. And then yet another, hopefully there is another resistance gene in the wings and ready to take over. Well, not a very sustainable situation, especially as a breeder starts to run out of R genes. So this search to replace this type of scenario is very important. So the different breeding strategies that are being used, certainly the single R gene variety is still something that is being employed. There are multi-line blends of cultivars or lines, each of which has a different R gene, and they're combined together in the seed that a farmer would grow in the same field. And the idea being that having different R genes, it's harder for the pathogen population to overcome all of those. A gene pyramid is similar, except that the R genes are within the same plant. Backcrossing is moving the R genes from unadapted or a wild relative or landrace into modern germplasm, often by marker-assisted selection. And then finally, genetic engineering or gene editing. I'll show a couple of examples of that. So some considerations for breeding. First of all, resistance has to be identified either within the crop species, that would be ideal, or a close relative. The close relative is sometimes problematic. There might be barriers in fertility or different ploidy levels, etc. But I think it's the importance of germplasm banks can't be overstated. And we have a great example on our campus here of the USDA backup germplasm storage. There are 500,000 successions there, and mostly unexplored. So the reservoir of resistance genes is something we count on, and that breeders often go to for searching for new genes. And then the disease triangle is manipulated to evaluate host resistance. So either by having a disease nursery in the field or in a controlled environment to screen for resistance, using a virulent pathogen, a favorable environment, for example, using misting in a greenhouse or whatever needs to be done to favor disease. Unfortunately, in the field, the occurrence of natural infection is often unreliable or non-uniform. So it can be a real problem if you get diseases in some years or some locations, but not every year. What most effective breeders have done is to develop a close relationship with a plant pathologist, because it really takes specialized facilities and skills to develop quantities of pathogen for inoculation, to investigate resistance mechanisms, to help evaluate resistance. So fortunately, most of our land grant institutions and private companies as well do employ plant pathologists to help this process. So durable resistance, that's the goal. Resistance that remains effective in a widely grown cultivar for a long time, even in a favorable environment. Durability of our genes depends on the evolutionary rate of the pathogen, the size of the population of the pathogen, and the particular R gene mechanism. Now, the rust diseases are probably the classic example of the use of R genes, and Andrew mentioned this morning with stem rust, how stem rust resistance gene, I think it was SR31, was effective for so many decades until it was overcome. And we have now various races of, or variations of the race of pathogen in Africa that has overcome that resistance. And there's leaf rust and stripe rust, all important in different parts of the world. Okay, transgenic, I'm going to come back to stripe rust, but transgenic disease resistance. Maybe the best example is resistance to ring spot virus in papaya. Some say that it's responsible for saving the papaya industry in Hawaii. It's been very successful. I think it's been grown since the late 90s. There are other examples of transgenics which have been produced, controlling virus, fungal, and bacterial diseases, but very few have been released. And there's a number of reasons for this. One of them, of course, is how difficult it is to get a transgenic variety produced. I think the estimates are $20 to $100 million for a new transgenic to go through all the regulatory procedures. Public concerns about biosafety, whether justified or not, that's one of the factors involved. Sometimes unwanted pleiotropic effects, so that a gene that provides resistance may also be responsible for reduced yield or some other important trait. And then perceived insufficient commercial benefit. If there are pesticides that are reasonably effective and available, then the benefit of investing in a transgenic is less. So we have at least one good example, but what is really gaining attention now is gene editing, especially using CRISPR-Cas9, although there are other systems as well. So I'm sure most in this room are familiar with the fact that gene editing empowers precise DNA changes, efficient, low cost. You don't have the foreign DNA insertion in the final product. And so some of the concerns with transgenics would be relieved. And correspondingly, the regulatory requirements may be simplified, thus reducing the cost of commercialization, so that it may even be possible for an organization like CSU to commercialize and patent a gene-edited product. There are some examples of gene editing for disease resistance in wheat, rice, cucumber, probably others as well. A nice thing about the technology with wheat, which is a polyploid, is that you can use, you can target all copies of the gene at once. So you can knock out, for example, if that is your goal, all of the gene copies rather than to do them one at a time. Another application of Cas9 which is kind of interesting, is not to use it to manipulate the plant DNA, but to detect and destroy invading pathogens. That was the original function of Cas9 in bacterial systems. And so if you could apply Cas9 enzyme to detect invading virus and degrade it, that could be an interesting application of the technology. Okay, now I'd like to give an example from Colorado. This is largely information from Scott Haley. So stripe rust is the particular rust that is important here, Coccinia stripiformis, and the picture indicates why stripe rust is an appropriate name, also called yellow rust. The cool night temperatures here favor stripe rust over the other rust. So really we don't see much in the way of stem rust or leaf rust. This is a relatively recent problem, only been a breeding objective since 2001. There was an important race change in 2010 and then 2012. And this follows the original race change in 2000-2001. So prior to 2001, it was something that didn't show up often at all, wasn't worthy of including in a breeding program. But then we had the race change, it became serious in certain years, and then again, and then a third time as well. So it can be hard keeping up with these changes. The keys for the problem in Colorado are moisture in Texas and Oklahoma, and precipitation here in May. So rust is a disease where the spores blow up from other states. So if there was poor development in Texas and Oklahoma, we probably wouldn't see much here. But reconnaissance is very important with rust disease so that we start getting reports early in spring and get prepared for when it might arrive here. So stripe rust is evaluated both on infection type, shown up above and scored on a 0 to 9 system, and then severity, what percentage of a leaf area is affected. And there's side by side of very susceptible and resistant variety. So you can see why yellow rust is also a name for this disease. It really turns a field golden yellow. So this is a graph of Scott's notes on stripe rust. So every time he scored a plot, and he has thousands of plots every year, every time he scored a plot for having stripe rust. And those are years across the bottom. So you can see that some years it warranted, a couple of thousand notes of his, sometimes up to 10,000 notes. In other years, practically none. So this might be a good thing for a farmer that the stripe rust doesn't occur every year. Not a great thing for a selection environment, because a breeder would like to see uniform selection pressure that one could select for every year. Anyway, it just shows some of the problem. So to deal with that, well, let me first say the breeding approaches start with the crossing. So Scott monitors closely regional and international nurseries for new sources of resistance, and then field selection. It has worked well here when the opportunity arises, but as I've shown, it doesn't always arise. So Scott relies heavily on collaborators in different states. There are nurseries in Texas, Kansas, and Washington State, where they reliably get stripe rust infections. So he sends hundreds of lines to these locations every year. So the importance of regional collaborations is really important here. And then greenhouse screening is another option that works for some diseases, not for this one. It's very finicky for temperature and humidity, and it's been very difficult to get uniform inoculation. So breeding approaches include marker-assisted selection for known genes, marker-assisted backcrossing to pyramid different genes in the same period, preliminary line and doubled haploid screening, and then some newer tools to leverage data. This allows Scott to make predictions even in years without field data. So things like genome-wide association studies, GWAS, and genomic selection. So as an example, here's the GWAS. So for those of you not familiar with this, the chromosomes of wheat are shown across the bottom, and then the significance of a particular marker is on the y-axis. And every dot indicates a DNA marker, which is evaluated for its association with a disease in this case. The higher the dot, the more significant that marker is associated with the disease. Oop. So in this panel, this was almost 800 individuals in Scott's breeding program. YR17 is one of the R genes associated with resistance, but it's combined with the quantitative resistance. It's out of a variety called TAM111, and it also has a strong association. So in this case, it's an R gene plus an apparent quantitative resistance involved in the breeding program. A different panel indicated that same YR17 being important, but then a few other QTLs that have so far been uncharacterized. So this is good. It's showing that resistance in the program is not limited to a single gene or a couple of major genes, but there are various loci involved. And then another one on 61, high level of significance, had previously been unreported. Okay, so current status of stripe rust resistance in Scott's program. The most resistant varieties are known as Canvas and Guardian. Isn't it strange how many things are named Canvas on this campus? Okay, sorry about that. So those two most resistant have pyramided, YR5 and YR15. There are other resistant lines that combine the YR17 and the TAM111 genes. He continues to cross parents with different sources of resistance, evaluate resistance here when possible, otherwise using the out-of-state nurseries, and refine predictions based on DNA markers, so that even in years when there's poor rust-free development here, or in those other states as well, based on DNA marker patterns, you can predict with an accuracy of maybe 0.5 what the resistance level would be. Okay, so I'm going to shift into insect resistance. And those are three of the problems, which are due to insects. Two are insects per se. The third one, barley yellowport virus, indicates that insects are also important because they're vectors for disease, especially viral diseases. The use of broad-spectrum insecticides is expected to decline because of resistance developing in insects, environmental and human health concerns, and the high cost of commercializing new products. And so there's a lot of emphasis continuing on developing resistance as an effective strategy. Looking at transgenics, Bt transgenes have been effective, but in a limited number of crops. Corn and cotton are the big ones in this country, with 80% or more of the area planted to Bt varieties. Around the world, Bt eggplant has been commercialized in Bangladesh and very effective. And there are efforts underway in other crops as well. There's attention being given to engineering the toxins. So for example, to combine the active domains of different Bt genes into the same gene construct. And then other protein toxins are being researched. Spider and scorpion venom, other types of inhibitors of insect function. So that's an active area of research. Consequences of widespread use of Bt genes. Kind of interesting, one of the benefits of widespread use of Bt genes is that some insects have virtually disappeared. European corn borer, for example, in the upper Midwest used to be an important problem, but the populations are so low that not only adopters of Bt technology have benefited, but non-adopters, including organic growers who raise corn in the same area, have also benefited. There has been field resistance developed in several species and regions. One of the ones that's a concern here is western corn rootworm in Iowa and Illinois. It's considered that resistance can be minimized through resistance management strategies, so using refuge of non-Bt varieties or stacking multiple Bt genes. So it's certainly a concern, but I think one that is manageable with appropriate stewardship. And then these are some ideas from that Angela Douglas review paper, promising opportunities for insect resistance. And one is engineering plant secondary metabolism. There are considered to be, I think, something like 200,000 different secondary metabolites in the plant kingdom, few of which have actually been investigated. So looking for new sources of toxic or insect deterrent chemicals. Crop wild relatives are considered to have a greater diversity of these metabolites than the crop domesticates. One example is 2 genes for terpene synthesis were transferred from a wild to a cultivated tomato and provided protection against whitefly and tomato hornworm. Harnessing plant immunity, just like there are R genes in the plant and pathogen system, there are also R genes involved with insects. So one example is soybean aphid and soybeans with resistance conferred by the so-called RAG genes. So understanding better the mechanism underlying these R gene interactions will facilitate their better use. And then the third area she mentions is managing the insect microbiome. Well, we hear about microbiomes in everything, from human health to animal production, plant breeding, and now in insects as well. So either target the microbes directly with antimicrobial peptides, for example, by sprays in the field, or actually engineer microbial communities that are ingested by insects to deliver insecticidal genes, such as RNAi. So actually, topics that I had never considered before, but it makes sense that microbes affect the insect-plant interaction just like everything else in the biosphere. So the example from Colorado that I'll give is the wheat stem sawfly. And I acknowledge Scott. I should also have acknowledged Frank Piers, our entomologist, because he's been involved with this insect for a lot of years as well. So this is a serious and expanding problem in wheat production. It had been confined to the northern spring wheat region, but then started appearing in Montana winter wheat, moved down into Wyoming, and eventually to Colorado in 2011. I believe it's now also appeared in Kansas and Nebraska, and has really been attracting a lot of attention as a current and future need for wheat breeders to focus on. So the nature of the damage, the insect lays its eggs in the stem, the larvae tunnel through the stem, inhibiting translocation, eventually cutting the stem, causing lodging so that harvesting is difficult. One side effect, it affects crop residue persistence. So you don't get the benefit of the stubble capturing moisture in the field. So for management, insecticides are not effective. Cropping can be partially effective as far as rotating crops. Host plant resistance, and the trait that is the leading trait and the one that has been most associated with resistance is the solid stem trait. So that you have a pith that inhibits either the laying of the eggs or the tunneling of the larvae. So this is 2011, the sweetnet showing a lot of adult flies there. Ripper was a variety that was very badly affected. Maybe you can see all the stems lying on the ground. And then Hatcher, another Colorado variety, not nearly as effective. So there is variation within the breeding program. These are results of the sawfly surveys. The epicenter seems like it's around New Raymer in Weld County, which is where one of those orange dots is in 2012. And it's continued to be a very bad problem there, but is spread every year. So the number of fields with a higher level of damage has increased. This was 2018. So maybe fewer fields with infestation, but the fields that were infested had a higher level of damage. So it looks like a problem that's here to stay. It's not going to go away anytime soon. How about those Montana varieties? Because they've been selecting up there for quite a few years. So Scott has grown these Montana winter wheats in his trials and found good levels of cutting resistance, reduced cutting, but also yield penalties. So up to 40% in some varieties. So using these other varieties here is not going to fly. There really needs to be a concerted effort to incorporate the resistance in Colorado adapted lines. So the breeding approaches Scott has used are double haploid breeding. This is a technique to accelerate the time in which that it takes to develop lines. Repeated phenotypic selection in segregating generations. Crossing elite lines, so his leading Colorado lines, crossing them with solid stem donors. Rapidly developing lines to evaluate under sawfly pressure. Then identifying the lines with potential increasing seed, and then recycling the lines back into the crossing program. So hopefully having a continually increasing level of resistance. So here's what cross sections of a stem might look like for different levels of stem solidness. The method that's used is to cut stems at the soil surface and then assess on a 1 to 5 scale over 5 internodes, so that there would be a total possible score of 25 for a given plant. So a 0 to 25 scale. Scott has used principally Judy, Bear Paw, Warhorse, and MTS line in his crossing to adapted parents here. He's used a selected bulk breeding method where he will do a back cross of the solid stem source to a local variety. What's shown here is bird, increasing and bulking the harvested plants, the selected plants, scoring on this 0 to 25 scale. So in that third box, you can see he advanced everything that had a composite score of 15 or greater. On the next cycle, he wanted to see things that were at least 20. And then line selection, preliminary line testing, and finally released. So just an example of the numbers of lines, 264 lines grown in 2015, 134 lines selected. They were scored for solidness in the field and also assayed for DNA markers. There is a major locus, it seems, connected to this trait on 3BL. So every year there was some reduction in number of selected lines, so that by 2018 it was down to 4 that seemed to have acceptable resistance and agronomic traits. And here's some of the data. This is showing Bird and Denali. Those are released lines, so those are checks of high yielded lines. And then the CO lines are the candidates for release for sawfly resistance. So you can see on the far right that the stem cutting is much reduced in the candidate lines. The solidness is much increased, but the yield in some cases has suffered. Not every year, but in some years at least. So that's an important consideration, that there can't be a yield penalty. So this is what it looks like in the field. The variety that is just being released this month, or next month, maybe, September, Fortify SF. SF standing for sawfly. So that's the first Colorado sawfly resistant release. Not bad, considering that the first appearance of the insect was 2011. So to actually have a released variety ready for farmers to start growing, at least to grow the foundation seed in 2019, is respectable progress, very good progress. So the Fortify, on the bottom photo, Fortify is on the left. The cutting percentage was 20% compared to Canvas on the right, 85%. So a major difference there. So I would think farmers that have had this trait or this insect show up in their area would be very interested in this variety. Okay, this is a GWAS of sawfly resistance showing the 3BL locus, very important. But there are other loci as well. So Scott is tracking these and trying to enrich his materials for these other loci as well, so that all the eggs aren't in that one basket. Okay, closing thoughts on wheat stem sawfly resistance group. So, deployment of semi-solid and solid cultivars. So, yeah, one thing I should say is that this released variety, Fortify, is not a super solid, it's a semi-solid. It was considered a compromise between deterring the insect and allowing reasonable yield as well. So what level of yield drag is acceptable? For a farmer that has had this problem year after year, some amount of yield drag would certainly be acceptable for that farmer, maybe not for a farmer that hasn't had the problem yet. Integration with sulfide population forecasting, if there could be some way of forecasting how serious the problem was going to be, it could influence a farmer's decision. And is the semi-solidness sufficient under a heavy infestation? Resistant sources, diversification, durability, effectiveness, stacking, those are all principles that Scott is following. One thing that's interesting, there's a stem rust resistance gene, SR2, which seems to also bring with it resistance to wheat stem sawfly. I'm not sure if it's completely determined whether it's just close linkage or whether it's the gene itself that's affecting both traits. And then there's a Turkish landrace that has 3 new QTLs for resistance. So again, pointing out the importance of germplasm collections. Is there resistance that's non-solid stem based? That might be another avenue to pursue. Okay, so another point I want to make, and this was in the review article by Nelson, is the importance of diversity, not just in a given variety, or not just on one farmer's field, but across a whole agricultural landscape. So it could be, stacking or pyramiding genes within a variety, but then a farmer growing several varieties, not just a single variety across 2,000 acres. As well as growing different crops, not just wheat, not just corn, but trying to integrate a rotation of systems so that across the whole agricultural landscape, we have an environment that is less conducive to rapid evolution of pathogens and insects. Okay, so final slide, just some conclusions and perspectives. The spread of pathogens and insect pests and their ongoing evolution make resistance breeding challenging. Certainly with climate change, we can expect that this might even accelerate. Breeding for resistance must occur together with selection for yield, quality, and other traits. So it's not just something that a breeder can focus on completely, but has to keep the whole package in mind. More durable resistance will place greater emphasis on quantitative rather than R gene resistance. Genomic tools, including high throughput DNA markers and gene editing, will empower genetic analysis and crop improvement. And finally, diversity at all levels of agroecosystems will promote durable resistance. Okay, and with that, I would welcome any questions you have. Thanks for your attention. Yes?
My friend Jack Tuick, who's a plant breeder at the University of Illinois, told me recently that he believes that breeding for resistance tends to emphasize diseases rather than insect pests because it's easier to work with. Can you agree with that?
I think so, yes. I mean, I think getting large batches of inocula that you could do artificial inoculation tends to be quite a bit easier than insects.
Do you think entomologists should help and make that easier for you?
Frank, you're leaving too early. Yes, and in some cases they've been effective. I think there are good examples of European corn borer and fall armyworm are being raised artificially for inoculation. But apparently this wheat stem sawfly is a very difficult one. So I think probably there are more similarities among pathogens as far as conditions and methods for increasing inoculum compared to insects that all have their very specific requirements. Yeah. Yes.
There's rumors out of Kansas that there's a new race of striped rust, 2019.
Oh,
Do you have any feel for how like Canvas might hold up against this new race?
oh, I don't know. I'm not sure. When Scott and I discussed that, he didn't mention this new race, but has that apparently overcome the resistance in some of the current varieties?
Some of the K-State varieties. I think the Wolf is trying to confirm how, if it is a new race.
Uh-huh. Well, certainly it argues for having multiple genes in every variety and not just depend on one or a couple. I guess there's the Canvas has the wire 5 plus 15, which has had a strong level of resistance. So hopefully if the new race overcomes one of those, it won't overcome both, at least not immediately. Okay, Todd.
How much of the yield drag in the solid system variety, or that was observed when you bring the Montana varieties to Colorado, was due to adaptation versus how much is due to the solid system trait itself? Was there a yield drag from that?
Yes, part of it is just adaptation. You know, it's a very different temperature environment down here. But apparently the solid stem trait itself, I mean, one thing is just it takes photosynthate to make a solid stem, right? But there seems to be something else involved, and I don't know the details about that. But yeah, so that is why Scott and Frank decided on the semi-solid rather than the really hard solid as the best compromise. Okay, thank you very much.
Okay, we have coffee, beverages, and some cookies, so help yourself, and we'll have reconvene at 4 for our last talk, and then we'll have a mixer after that. So.
Diversity.